Hello, in this video we're going to be talking about potassium shaker channels. We're going to be talking about a few things. The first thing that we're going to be talking about is their role in the repolarization of the cellular membrane. We'll also be talking about how they impact the duration of the falling phase of an action potential. We'll talk about their contributions to the firing rate. We'll talk about the structure of the shaker channels, the functions of the shaker channels, and how that structure impacts the shaker channel functions. So let's get started. Here we're looking at a diagram of an action potential. We have -70, 0, and 60 millivolts. So I'm just drawing out a really quick diagram of this action potential. We're mostly focused on 4 different parts. So the first part is when the threshold is reached, those sodium channels all open, those voltage gated sodium channels open, and then all that sodium flows into the cell. The second part is the peak, where those sodium channels are inactivated through that backdoor mechanism. Those potassium channels open, and then the falling phase, where the potassium flows out of the membrane, out of the cell, repolarizing the membrane. And then the last is the hyperpolarization, when the potassium channels close and the sodium channels deactivate, ready to activate again for another action potential. Now, if we zoom into the hyperpolarization phase, this is when the termination of the stimulus occurs. So this is when there's that lack of depolarization. We're back at the resting membrane potential. And sometimes there's an inactivation of potassium channels instead of them actually being closed. Sometimes it actually will happen before complete repolarization. Now we're going to get into the structure of these potassium channels. So each shaker potassium channel has 4 alpha subunits. And each of these alpha subunits have 6 protein helices. So I'm just drawing some lines to denote those protein helices. And these helices are transmembrane. That means that they go through the membrane. So one end of the helice is on the outside of the membrane. The underside of the helix is on the inside of the membrane. And these 6 helices make up 3 domains per alpha subunit. So the first is our pore-forming domain. The second is our voltage-sensitive or voltage-forming domain. And the third, which we'll talk about later, is a little tricky. This is our channel gate domain. So here I'm just drawing out those 6 protein helices again. And on the 5th and the 6th helix, we have a P-loop. And this P-loop is also functions as our sensitivity filter. So this is what makes sure that only potassium ions are going into the pore. Next, we have 4 other protein helices. So these are protein helices 1 through 4. And I'm just showing that sodium can't go through that sensitivity filter. So 1 through 4 is our voltage sensing domain. And the 4th is special because the 4th has positively charged amino acid residue in it. And basically, when the cell membrane potential changes, those positively charged proteins will actually move according to the difference of charge. And we'll talk a little bit more about that later. And then on the 6th helix, we have the C-terminus, which is a carboxyl group at the end of a chain of amino acids. And then we have our N-terminus, which is an amine group at the end of a chain of amino acids. So I'm just going to get a little bit more in detail here. So I'm drawing out my 6 protein helices. And I'm actually drawing a little corkscrew in here to show that even though I'm drawing them as boxes, they are actual helixes. And I'm drawing that pore loop, again, our sensitivity filter, and in between each of these helices is an amino acid chain that binds all of our proteins together. On our sixth helix, we have our carboxyl, and on our first, we have our amine. So here's just a little model that I made of our 6 helices of our alpha subunit, and you can see those last 2 helices are making our pore-forming domain, and our last 4 are our voltage sensing domain. And then we have our little p-loop connected right there. We also, of course, have our N-terminus with our amine group, and our C-terminus with our carboxyl group. Now, if you look at the shaker channel from the top or the bottom, you'll see those last 2 helices, that 5th and 6th helix, kind of connecting to each other to create a hole. That's actually what creates the pore. And you can see those P loops, those sensitivity filters that are inside the pore, and those are what are, filtering what ions are able to go through. And then connected to those 5th and 6th helices, we have our 1 through 5 voltage gated, 1 through 6 voltage gated domain. So there we have our pore forming domain with our 5th and 6th helices. And then we have our P loops. And the P loops function, it's thought that they function by basically dehydrating the potassium ions to actually make them smaller than the other ions, which then will allow them to go through the pore. A lot of the information that we have on shaker channels is actually based on prokaryotic organisms. And so a lot of what we have is based on experiments where we will do certain things to certain structures and see how they react. And so this is just another model that I made showing that top view, and you can see those pore filters letting the potassium through, but not letting the sodium in. And then on the bottom, you can see our different terminuses. We have our N-terminus and our C-terminus. And then this right here is a beta subunit. And I'll talk a little bit more about that later, but I'm just showing that the beta subunits, depending on the type, can bind to either that C-terminus or that N-terminus. Now we're going to be talking about channel gating. Channel gating is when there's actually a conformational change of those pore-forming helices, and that results in the opening and the closing of the actual pore. We don't know the mechanism. That's what the question mark is for. So I'm just drawing our main voltage gating helix. So this is our 4th helix, and it's filled with positively charged amino acids. So as we know, the inside of the cell is typically more negatively charged than the outside. And what will actually happen is, as depolarization occurs, those positively charged amino acids are going to move to the side that is, of course, more negative. Positive is attracted to negative. Now, this leads to a change in the structure of the pore. This is what opens and closes the pore. So as the pore is closed, you can see the inside of the cells more negative, so those positively charged amino acids are actually outside of the channel. They're sort of inside of the cell. Then depolarization occurs, and those positively charged amino acids move, and they move to the outside of the cell. And so here I'm just showing the actual channel pore. Here it's closed. Depolarization occurs. Those amino acids move. The pore opens, and potassium is able to flow outside of the membrane. Now, how this actually happens, what mechanism is occurring to connect these two events is still a mystery. Now we're going to be talking about inactivation. So similar to sodium channels, shaker channels can also inactivate, which stops the potassium efflux without the removal of the stimulus, without the complete repolarization of the cell. So there's 2 types, there's N-type and there's C-type. The N-type inactivation is also called the ball and chain inactivation, and this is the fast inactivation. It's very fast. So I'm just drawing our protein helices, all 6 of them, and I'm drawing our N-terminus at the end of our amino acid chain. And then there's our pore, and I'm just labeling our helices, 1 through 6. Now, the pore is open and potassium can travel through the pore, but this amine group can move to the pore, and this will actually, as the name implies, function as a ball attached to a chain, an amino acid chain. And this will actually basically plug the hole of the pore, preventing any potassium from going through. And this is a very quick method of inactivation. And it's impacted by the chain length. This is really important, and I'll talk about it. So just to talk a little bit more about N-type inactivation, this decreases the duration of the falling phase. So because it's so quick, we have our normal action potential here, and we have like an extended falling phase where the potassium is going out. But with a shaker channel that has N-type inactivation, you can see that the falling phase is much shorter. It's a much shorter duration. And that's because since that ball is able to really quickly block the pore, the potassium has to leave really quickly. So now we're going to talk about C-type inactivation, which is our slow type of inactivation. The mechanism of this is still unknown, but we do know that it has to do with conformational changes to that channel pore. So basically that channel pore, instead of closing all at once, it slowly closes over the period of time of the falling phase. So here again, we have our typical action potential with our typical falling phase. And we have our potassium leaving the cell. However, a shaker channel with C-type inactivation is going to have a much longer falling phase. And that's because once that peak is reached, the pore kind of closes slowly, theoretically, or closes gradually, which causes a slow efflux of potassium out of the cell. And this increases the action potential duration, which decreases the overall rate of firing. We also have beta subunits. These beta subunits are not well understood, but we do know that for every alpha subunit, we have one corresponding beta subunit. So here I am just drawing out our protein helices again. We have our 2 pore-forming helices and our 4 voltage-sensing helices. That's our sensitivity filter, our amine group on that 6th helice, and our carboxyl group on that 1st helice. Now, we have different types of beta subunits, and they can either bind to the N or the C terminus. And depending on which terminus they bind to will affect the function of our different modes of inactivation. So a second beta subunit, B2, will bind to that C terminus, and that will impact conformational change. It will impact our C-type inactivation, whereas B1 binds to our amine group, and that will impact our voltage sensing ability, and any mutations to these beta subunits can result in changes in our firing rates. Now we're going to talk about abnormal structural changes. Any change in the structure that's abnormal could cause a change in the overall firing rate. This could be due to changes in inactivation. An increase would lead to overexcitation, which is linked to epilepsy and Fragile X syndrome, whereas a decrease would be underexcitation. And there's a few reasons for this. One is if there's any changes or mutations to the beta subunits, that could impact the channel's ability to sense changes in the voltage of the membrane. or it could be due to shortened amino acid chains for the N-terminus. If the amino acid chain is too short, the amine group is not able to reach the pore. So, in summary, shaker channels have forms of inactivation similar to sodium channels. Their inactivation can increase or decrease the duration of the falling phase, which can change the overall firing rate of the cell. The structure of the shaker channel is very important because it influences the function of the shaker channel, which influences the rate of activation and the firing rate. And there's still lots unknown. We don't know a lot about the underlying mechanisms of all of these different functions. Thank you so much for watching.